A very good morning to all of you all attending this session. I hope I'm audible. Can I just have a quick response on the chat box? Yes. Okay. Thank you all of you all. We were just waiting for some more participants to join, as some of them were finding it difficult to join the session. So once again, good morning to all of you all, and welcome to the Inspire Camp, the day 2 of the Inspire Camp. Most of you all must have attended day 1 on 12th of December. Some of you all must be new as well. So basically, Inspire Camp is an initiative which is taken by ISA and managed under the Department of Science and Technology to hunt for new talents in science, right? So basically, when we talk about career, when we talk about career options, we get into engineering, we get into medical, but there is a lot of importance regarding pure science as well. Inspire Camp is an initiative to make you understand the importance of research by inviting eminent scientists from ISER itself, who will themselves be guiding you through their path, through their research, for a better understanding of what research is all about. As a teacher, I have personally seen students speaking statements like, "मुझे तो research में interest ही नहीं है," and that's exactly the point where I say to myself, "इसको research के बारे में पता ही नहीं," right? So I'm sure Inspire Camp takes a big step towards it, so that you stay in the country, prevent yourself from brain draining yourself, and get various fields of science to some particular level in research. Inspire Camp 2021 wouldn't have been possible without the initiative taken by Prabhu Desai Charitable Trust, which is a trust working at various sectors across Pune and Ratnagiri. Along with educational institutes, the trust also promotes or extends help towards health sector, hospitals, ashram shalas, and invites dignitaries, scientists, and business tycoons for such inspirational events. So I extend my heartfelt gratitude to Prabhu Desai Charitable Trust. Today, we'll be having two speakers from 10:30 to 11:45, and again from 12 to 1:15. And without further delay, let me introduce to you today's speaker, Professor B. Nagaraj from ISER, Pune. Professor Nagaraj is an associate professor at ISER, Pune. After completing his BSc and MSc in microbiology from Mumbai University, Sir has completed his PhD from Cancer Research Institute, Tata Memorial, associated to Mumbai University again. He has 19 publications on his name and has been invited to 14 research talks. He holds the position of postdoctoral research associate in molecular and cellular pharmacology at University of Miami School of Medicine at Florida, postdoctoral research associate, cardiovascular research center, University of Virginia, research scientist, cardiovascular research center, University of Virginia, associate professor, Department of Biology at ISER Pune, adjunct faculty, Department of Biology at Pune University. He has been awarded by the Wellcome Trust DBT Alliance Senior Fellowship, India, Ramalinga Swamy Fellowship, Department of Biotechnology, India, Ruth Christian National Research Service Award, NRSA Fellowship, Gordon Research Conference Gradient Sensing Migration Travel Award, a Senior Research Fellowship, CISR, Government of India, and his 19 publications in various fields. You'll be listening from him about one of the topics he's going to talk today. So I welcome Professor Nagaraj and hand over the session to him. Over to you, sir.
Thank you. Thank you so much. Can you hear me? Are you able to hear me?
Yes, sir.
Okay,
Yes, sir.
okay. Thanks again for everybody who is here. And it's always wonderful to be part of an Inspire camp. For me, it is one of the most exciting things I look forward to. It's an opportunity to interact with many students from across the country. And I'm glad that these camps are, back in session. And even though we are doing this virtually, I think the opportunity to interact with students like this and for students to listen in is something that is always very exciting. Okay, so um, uh, thanks again to the Prabhu Desai Trust for supporting this. I think it's really commendable that they think this is worth investing time and money towards. And the Inspire camps are supposed to give many students like you the opportunity to interact with scientists, hear a little bit about their work, and be excited by the kind of things that we are doing, right? So my hope also in the next maybe hour or so is to try and get you excited about something that we study in the lab, and I'm going to kind of build a story about why we study what we study, and then tell you a little bit about some things that we do actively in the lab to give you a sense of what kind of experimental work we also conduct, right? So I'm going to pull up a presentation. Let me just share this screen. One second. Are you able to see the screen?
Yes, sir. We are able to see it. Okay.
Well, I'm just going to try and my video so I can see this and see if I can get a pointer up as well. Okay, I can bring up the mouse. Okay, I can't see the mouse for some reason. I don't know whether you are able to. Okay, let me do this without the mouse. That's okay. So you you guys can all see the slide as well as can hear me clearly, right? Okay. Yeah, so what we'll do is the way I would like to do this is that I'm going to tell you about what I'm going to talk about. You're welcome to put in questions in the chat box, as they occur to you, during the talk. At the end, we'll try and I'll go through those questions and answer them as well. And the hope is that we can take questions in person as well at the end. Okay. So the title of my talk is the matrix of multicellular life. Some of you may be wondering what this is and what it means and all of that. And that's perfectly fine. The intent here is to kind of try and give you a sense of what is it that, I mean when I say, matrix of multicellular life. Right. The second corollary to my question is the title says matrix of multicellular life and says how cell adhesion regulates cell function. Right. So most of us know what cells are, but you also probably know what I mean when I say cell function. What you probably don't know is what exactly is cell adhesion. right? And that's one of the things I'm going to try and introduce to you today. And the reason I'm particularly keen to tell you about this is that's what we in the lab study. So my lab focuses on cell adhesion. My lab is located here in ISER Pune, and some of you may be from Pune, and you may have come to the campus, or because of the pandemic over the last few years, 2 years or so. I think if you haven't been to the campus, come to the campus and see us at some point. It's a really beautiful campus, green campus located in the middle of the city, and we have both undergraduate and PhD students. We have about 2,000, students who live and study on campus, right? Coming to the question that I was asking you, one sec. My slide refuses to move. Okay. Okay. So the question that I raised earlier is, what exactly does the term cell adhesion mean to you, right? And cells, we are all familiar with, and you guys all know what cells are. The question is, what does this term adhesion mean, right? And if you go look up the dictionary, the way the term adhesion is defined is the ability to stick or become attached to something, right? So cell adhesion essentially translates into the ability of cells to actually stick and become attached to something. Now, that's a very interesting thought, because I don't know how many of you have considered this as to whether cells are actually sticky, right? Do you think that they can stick at all, right? And this is what we are going to explore today, right? And we are going to do this by addressing a set of questions, right? I'm going to try and address the question of, do cells stick? We'll try and ask, do they need to stick? What exactly is kind of advantage or disadvantage that we may have to have cells that are actually sticking to something? The question then is, what can they stick to, right? So do they stick to other cells? Do they stick to something else? Is an interesting question. Then the question is, if they do stick, how do they stick, right? And what is it, the mechanism that they use to do this? And finally, we will try and address what happens if they don't stick, right? If all this is true and cells need to do this, if they cannot stick, what exactly happens to cells in our body. So the human body is made up of many organs, which are themselves composed of tissues, and the tissues then make up cells, right? Are made up of cells. A good example of this is our brain, right? Right now, the fact that I am talking to you is made possible because of the thoughts that are coming from my brain, which also controls my hands, my mouth, what I'm saying, the language that I'm using. Everything is controlled by the brain. The fact that you guys are listening to this, at least maybe some of you are, is mediated also because of your brain, right? The fact that your brain is able to listen to or control your body to allow you to listen to what I'm saying, the fact that it is able to process what I'm saying and make sense of it, or not make sense of it, is all made possible by this kind of singular organ, right? And the brain has a complex structure, right? That's how the brain in real life looks like. And if you cut it and you look at it carefully, and this is an actual image of the brain, which is cut and a cross section is taken, you can see it's made up of billions of cells that are all put together nicely packed, and then have a very distinct architecture too, right? Because if you've seen a picture of the brain, and I showed you this picture now today, you immediately, even if I had not told you, you would probably have understood that this is the brain. And some of that is coming because of the architecture of that brain. The way it is folded, the shape it has, all of this. But imagine all of this is made up of individual small cells, right? And isn't it remarkable that a complex architecture of a tissue and an organ is created very reproducibly, right? All our brains look kind of the same, right? There might be smaller differences in how things are organized. Some of us are more interested in music and some are less. But they're all made up of cells, and the cells come together to create this complex structure, right? So this is really fascinating to see, right? And that's a zoomed in image of the cells, the neurons in a real brain, in a real human brain, right? And they are packed in a very dense manner to create that architecture, and they're working with each other to allow us to do everything that we are going to do this morning, allowing me to communicate, and for you to also understand what I'm communicating, right? So the brain in that sense is really the control center for so much that we do, right? And so the question becomes, now that we have thought about the brain, and we have thought about how these cells are nicely tightly packed with each other, it seems inevitable, if I raise this question to you now and ask, you tell me, if cells that have to be packed in a very compact organ, millions of cells have to be brought together, there has to be some cohesion, some stickiness to them, right? They have to be stuck together in some way that allows them not only to be packed as well as they are, but maybe talk to each other, and also acquire this very distinct architecture that we know is unique to that particular organ, right? Because a kidney looks very different from a brain, right? And if you go in and look at the kidney cell and the brain cell, what they are actually made up of is not very different. They may have certain properties that are different, but there are millions of cells in the kidney, there are millions of cells in the brain, but here the brain cells have a very distinct architecture for the organ that is the brain, and the kidney cells have a very distinct architecture for the organ that is the kidney. So cells are coming together and creating these complex architectures, in the process of development. So this is really, remarkable to see, right? And I'm going to try and show you a movie, before which we look at these set of images, which are essentially embryos, right? Because we all developed as embryos. So when we talk about a group of cells coming together to create something, I think the best example of these, are all of us, right? That we all began like a ball of cells, and these are embryos of different animals, and you have a dolphin and an elephant, and you can, after a certain age of the embryo, tell that the embryo belongs to a particular animal. But to begin with, we all began as a ball of cells, right? So 2-cell stage, 4-cell stage, 8-cell stage, and then we grew. And if you think about it, it's quite radical, in my opinion, that we start as a small ball of cells, and then grow to be these, wonderful, individualistic creatures, right? Each one of us having a personality, having interests, and all of that is governed. It all began, as a small group of cells, which are actually stuck to each other, right? So not only the complex organs, that I just told you about, the brain and the kidney, but even the beginning of life for all of us has been a group of cells that are stuck to each other, right? So that's the really interesting thing, that this ability of cells to come together and be stuck with each other is at the core of all life, right? And how life has evolved. I'm going to play a movie, and just watch this movie first. It's we are looking at the development of an embryo in an organism that is used very regularly in labs called the fruit fly. We've all seen the fruit fly. If you leave a banana out at home for too long, these flies, you will start seeing them, sitting on the ripening banana, right? And next time, if you can, see if you can catch hold of one of them. And even if you have a small magnifying glass, zoom in and look at them. That's the fly that I'm just showing you, right? And the embryos of these Drosophila of flies, Drosophila in general, has been used as a tool to study a lot of human development. Why I'm saying human development? Because what we learn in flies, a good amount of it actually translates into higher organisms as well. Flies also give us the opportunity to study some of these things and do experiments like we do here with embryos that is not possible to do with human embryos, right? So when you look at the embryo of the fruit fly, among the things that I'm hoping you will notice is how much change is happening as the embryo develops. You can see cells are all packed very nicely with each other, and they are moving in very distinct patterns, right? There is a small ridge or a cleft that is created on the left hand side. If you keep watching, you will see it happen. There you go, right? And then there is this movement that is taking place, and a lot of what's happening here has been very, very carefully studied. There are labs in IISER Pune who study, Drosophila development, and there have been Nobel Prizes awarded to the understanding of the development of the embryo here. But the reason I'm showing you this movie is to make the point that here is a group of cells in a fly that are actually in contact with each other and working with each other to develop patterns, and create movement in a developing embryo, which eventually becomes the complex organism that is the fly. The fly has wings, it has a liver, it has it has a gut, it has a heart. All these things are created by this group of cells, just as it is for us. Okay, that a ball of cells actually grows to become complex organisms like us and all other animals that we know of, right? So so when you think of this question now, right, which is whether cells can or need to stick to each other, the answer becomes more and more obvious that yes, it does look like this is something that's important, for cells and for complex organisms to develop. And another example that we all can actually see and feel, and which is one of the reasons I include it here, is the ability of cells to also contribute to our body's defense, right? And in this context also, the ability of the cells to adhere or stick to each other, just as it is important in development of organisms and tissues, it's important in processes of cellular defense, right? And when I say defense, the first line of defense, that's there for all of us, is something that is holding all of us together, right? Now pick up your your arm and look at your skin. Okay, just feel your skin, touch it, and this is your first line of defense, right? And think about it, right? That this is a tissue that's actually wrapping you up, right? That's wrapping your entire body, and is made up of cells, right? These are all cells that are stuck to each other, right? I don't know if it's ever happened to you that you burnt yourself, or if you go out for in the sun too long, you get sunburned, for example. And when you get sunburned, you will have a layer of cells that comes off over the next 2 or 3 days. And if you look at those cells, these are cells, right? Just as the kidney and the brain is made up of cells, your skin is made up of cells too. And these are coming together to create this very elaborate, intricate, and remarkable organ, okay, which does so much to protect us, right? And look at the properties that the skin has, okay. Among the things that it can do is, obviously, it can stretch. I can pull on this, and you can stretch your skin. It's able to stretch across your body, which means, as the size of your body increases, it grows with you. It has this remarkable ability to kind of resist water. Right, you take a bath, and your skin the water rolls off your skin, and it ensures that every time you go in the rain, you don't melt, right, because it is resistant to water, and it has the ability that if, for example, you get scratched, or you get cut, that it heals. Right? I mean, think about it. What a remarkable set of capabilities to have. If I were to design something, that protects something that is important, I would like to have all these properties, right? And that's the remarkable thing about the skin. And it's made up of cells that have been brought together and can stick together. And the ability of these cells to stick to each other is hugely important for the skin cells. Now, this is an example of an experiment that we do very regularly in the lab, and this movie is taken from somebody else, but we are taking a skin fibroblast cell and we are putting it on a dish, and we are watching what happens to the skin fibroblasts. Okay, so just watch what the cell does. Remember that begin with, it's a round cell, and the movie will play again, so you will get to see what the cell looks like eventually. And then when the movie goes back to its first frame, you will see how that cell began, right? So if you're taking a cell, you're putting it on a dish, and you're asking it to spread. And you can see the nucleus, you can see all the organelles that are moving inside, you can see vesicles that are moving, you can see organelles like the Golgi, you can see very nice protrusions that the cell membrane is making, right? This is like a giant balloon that is being stretched in all directions, right? And is getting bigger and bigger, right? Till the movie snaps and goes back to the first frame, and you will see how it began. Okay, that it began as a very tiny ball. There you go, right? And you can now see how dramatically it has stretched, right? And this is something that the cell is doing just because it has become adherent to a surface, right? So it became adherent to the surface, and now it's spreading, right? Now, the interesting thing here to consider is, for a cell, as I said, if you think about it as like a bag or a balloon, right? The cell needs to be able to attach and spread. Okay, I'll give you another example, and think about this, the thought that I just put into your head, which is, the cell, if it has to stretch, has to attach. Now, if you're a giant balloon and you are having to be stretched in all directions, think of what you will actually need to do. Okay. Similarly, think about this movie as well, right? Which is actually a movie that was shot a long time ago, 1950s or so. And this is a movie that shows you a cell that is actually a cell that protects us. Every time you get a cold, these cells actually come and fight the cold. Okay, and they're called macrophages. And this is a macrophage that is chasing the bacteria. I'm going to play the movie as I talk to you about it. So keep watching the movie, and then it will play again, and you will get to see it again. And the macrophage is again a giant balloon. Okay, and imagine the cell that we saw in the earlier video, which is now getting stretched. Okay, now here is a cell that is a giant balloon that's actually moving, right? And the remarkable thing about this cell is that this cell is able to chase this bacteria, right? I mean, we'll watch the movie again. Here we go, and you can see that the bacteria is wriggling away onto the left. The cell turns left. The bacteria turns right. The cell turns right. It doesn't give up, right? The cell somehow seems to know where the bacteria is. It's turning. It's making turns, and you can see it keeps chasing the bacteria till it eventually catches up with it and engulfs it, right? And this is one of the things that macrophages do to protect us, that they, go in and eat up bacteria in our body, and in doing so, they are able to protect us. But if you watch this movie carefully, among the things that the macrophage is doing, okay, I'll play it again, is this remarkable ability to move, right? The remarkable ability to go from one place to another and follow something as well, right? Now, for us, if you were, chasing somebody, and you're having to kind of follow where they go, there is a whole bunch of things that you have to kind of know and understand to be able to do it, right? You have to be able to see, you have to be able to know that if the person is going in this direction, the direction I have to turn is this. Now, it seems like a very simple thing, and even with our brain processes this information very, very quickly, so you can make those turns, right? But imagine a cell doing this, right? And among the thing that this cell has to do to be able to do this is it has to be able to attach, right? My favorite example is that of a caterpillar, and we've all probably seen a caterpillar move, and the caterpillar does something very interesting, right? That it attaches on the backside, it stretches, attaches in the front, pulls the back closer, right? And then again does this, and then pulls the back closer, right? So this ensures that the caterpillar is able to move, and the cells are doing more or less the same thing. There are places in the front of the cell when the cell is migrating in this particular direction that need to be adherent, and at the same time, the back of the cell has to be detached to allow the cell to kind of make these movements that we saw in the earlier movie and catch up with the bacteria eventually, right? And so this is also a good example of how movement is, or the ability of cells to do something is governed by, the fact that they are able to stick, right? So then the question becomes, okay, I get it. I understand that the cells have to stick. They have to stick to be able to make complex tissues. They need to stick to be able to, during development, they need to stick, to do this kind of migration that you just showed us, right? The question is, what can they stick to, right? And that's the next question we wanted to answer, right? So among the things that the cells can stick to is something called the matrix, right? So, I remember, maybe you guys are too young for this, but there is a set of movies called The Matrix, right? And which essentially, is about this network of, or the matrix as it is called, that surrounds everything and everybody, right? That idea is actually originated from this extracellular matrix, because this is the matrix that in physiology, in our bodies, exists in every single cell, tissue, and organ, right? It's something that the cells make, and that's the really remarkable thing about this, that the cells make this matrix, which then creates like a complex network or mesh in which the cells are sitting, and this matrix ensures that this cells come together and stay together and function together. Okay, the matrix. This is an actual image of the matrix that cells are made. The matrix here is in blue. And the cells here, which are these cancer cells that are making this matrix, is in orange or pink, right? So you can see that this is like a mesh, right, that is surrounding the cells, and the cells have made this mesh. They have contributed to this mesh. My favorite analogy to the matrix is actually is Maggie, right? And we've all had a bowl of Maggie. And I don't know if, like me, you've ever forgotten a bowl of Maggie in a bowl, right? And if you leave Maggie in a bowl, with vegetables and other things, after some time, it kind of solidifies and takes the shape of the bowl, right? So next time this happens, try it. Leave it in a small bowl, and then take the bowl and invert it and take out the bowl, right? And you will see this mesh that is holding all your vegetables in, and has a characteristic shape of that bowl, right? That's pretty much what the matrix is, right? If you consider all these vegetables to be cells, the matrix is this sticky mesh that surrounds them. But unlike the Maggie, this Maggie of life, of this extracellular matrix, is actually made by the cells, right? So the question then becomes, so what happens, how prevalent is this network, right? And a good example of this is, where you can take an organ like the heart here, and you can get rid of all the cells. I'm not going to tell you how you can do this. There is methodologies that are available that solubilize everything that is cellular inside. And when you do that, what you notice is you have like this white ghost of a heart, right? And this ghost of a heart is nothing but the extracellular matrix, right? That the cells have made, right? So this is like taking that bowl of Maggie, right, which has a particular shape, and removing all the vegetables from it, and the Maggie will still keep its shape, right? Because that architecture is what's keeping the cells where they are, right? And so the extracellular matrix that the cells make actually define this architecture, okay, actually define the architecture of tissues and organs that we see. So if you remove cells from the heart, what you're left with is the shape of the heart that is defined by this extracellular matrix. There are no cells, so it is kind of white in color here, but it's what's defining the shape, right? Now, as I showed you earlier, this Maggie is like a mesh, right? And it's made up of many different proteins. I'm listing 4 very important proteins here: collagen, elastin, fibronectin, fibrinogen, and there are many other proteins that all come together to make this mesh. Collagen is a very favorite, extracellular matrix protein because collagen, elastin are actually what's responsible for your skin, being as elastic as you are. I don't know if you have grandparents at home. I grew up with my grandmother, and I remember, as you grow older, your skin loses its elasticity, right? So if you live with a grandparent at home today, try this if you haven't done it already, probably, so you can go try and pull your skin and see how long it takes for the skin to get back. And you can try and gently pull their skin and see how long it takes, right? It will take much longer, simply because the collagen and elastin extracellular matrix with age change very dramatically. So this is another interesting point, right? Not only is the fact that the extracellular matrix is made by cells very unique, but because it is made up by cells, as the cells age, as your body ages, right, the extracellular matrix architecture, organization could also change, right? But this is the mesh that is created. The other interesting thing is that because you have so many different things, right? And this is like recipes we create in the kitchen, right? You can use the same haldi and ginger and coriander, but you can use them in different ratios to create very different tastes and flavors, right? Similarly, the extracellular matrix is something that is varying in its composition depending upon different tissues and organs. So if you look at the brain and you look at the kidney, and not only is their shape different, the cells that make the kidney and the brain different, but if you look at the composition of the extracellular matrix, it will also be different. So what is really interesting then is that the cells in the kidney seem to know their extracellular matrix, right? If they sit in a matrix, in an environment that they grow in, they know that environment. So if you take cells from the kidney and put them in the matrix that is coming from the brain, chances are they are going to first be able to tell that there is something that's not right. Okay. My favorite example of this is, many of us travel to go study abroad or even in different cities within India. And the moment you leave home and you go to a new place, it takes a while for you to get adjusted to the place. Over a period of time, you will get comfortable, being there. I grew up in Bombay, right? And I'm in Pune for the last maybe 10, 12 years. And I was abroad for about 10, 12 years. And each one was a very different experience. right? And it took a while to get used to each of these places, right? So cells feel the same way, and part of that is the environment, right? And this environment for them is the matrix. So you take cells from the kidney and you give them a new environment of the brain. It's the matrix that they are actually going to sense and say, oh, this doesn't look right or feel right, and they may take some time to get adjusted to the matrix that's coming from the brain. In many cases, they actually will not do well in that matrix, right? Which is one reason why cells from the kidney are never found in other organs, okay, unless and until, right, there is a disease condition, right? And I'm going to tell you at the end a little bit about the disease condition, which could lead to cells from, say, the liver, appearing in the lung, or from the kidney, appearing somewhere else in the body, right? But in normal tissues, this does not happen. Okay, so that's the matrix, and this is how the matrix looks like when you take them from different tissues, and they control a lot of different things. They control behavior of the cells, survival, development, migration, proliferation, shape of the cells, and cellular function. All is governed by the extracellular matrix. And this is one extracellular matrix, which is collagen that we are looking at, right? That's doing this. This then brings us to the all-important questions that along with the matrix, right, is there anything else that they can stick to? And, I said this earlier, but I'm repeating it again, that cells can also stick to each other. Okay. This is an example of a cell that actually lines your blood vessels, so it's on the inner lining of the blood vessel. There is a cell called the endothelial cell. And on the right, you can see, if you open that sheet of cells and look at them, the red is the boundary of each cell, the green is the nucleus there. But you can see that these cells are nicely packed with each other, right? And it's like a mosaic, right? And they make very, very nice, tight, interesting seals, between cells. So cells stick to each other, by my favorite example here is that of a zipper, right? And we all used a zipper, and it works really, remarkably well, allowing you to kind of close two edges, right? And the really nice thing about the zipper is it's reversible, right? So once you kind of zip up, you can zip down, right? Which means this two edges can be open, closed as required, right? And the cell junctions are also designed this way, that they can be controlled, so they can stick very firmly if required, they can be loose, they can detach if required, and so they are reversible structures, right? And this is made by a number of interesting molecules, right, that allow for cells to stick to each other, which is what we will come to next, saying that, okay, if cells can stick to things, and that something could be other cells, or that could be the extracellular matrix, what in the cell actually allows cells to stick, right? And for the cell's ability to bind to the extracellular matrix, they have very interesting receptors on the cell membrane, right? So the cell is like a bag, right? And this bag, is made up of a lipid membrane. Some of you may have read about this, that it's made up of lipids, which allows it to kind of fold and bend the way it wants. And in this, lipid membrane are receptors, right? And so the receptors go through the lipid membrane like this, right? So there is a part of the receptor that's outside, there is part of the receptor inside, things bind on the inside, things bind on the outside. And the receptors that bind the extracellular matrix are called integrins, right? A good example of how to think about integrins is, Velcro, right? And you guys may have all used Velcro. Shoes and other places these are used, right? The interesting thing about Velcro is, next time look at it carefully. It has a kind of a soft surface, and it has another surface which is actually made up of tiny, tiny hooks. If you haven't looked at it carefully, look at it again, right? And what happens when you press them too is that the two sides is that this fluffy surface, these hooks go and attach to, right? And that ensures that these two pieces are stuck to each other as long as you want them stuck, right? The nice thing about Velcro is that you can also rip them apart when you need. All you need is force, right? So you have to apply a certain amount of force and peel this, and it'll come apart, right? You stick it again, it will stick again. It's a reusable structure, right? And the attachment requires a certain amount of force to be applied. The detachment requires a certain amount of force to be applied. And that's kind of what the integrin extracellular matrix interaction also looks like. So imagine a cell membrane which has these hooks of the Velcro, and imagine the outside microenvironment, which is the extracellular matrix, being this fluffy side of the Velcro, right? And so every time the cell sticks, it will go bind and hold on, and this has the ability to kind of peel apart if required and come back again. It can go bind in a new place. If you're a cell like the cell that is chasing the macrophage that is chasing a bacteria, you want to be able to attach and detach, right? So if you attach and you stay attached, what's the point? You will not be able to move. You will not be able to change your shape, right? So this ability for these interactions to be reversible is very, very important. Okay, and this is another very important concept in cell biology and the biology of cells. There are a lot of things that cells do are reversible, right? So they can turn something on and then turn something off. They can attach and then they can detach, right? And this is a good example of that. This is how these structures look inside the cell, and you don't have to worry about it. But the only point it's trying to make is that what it binds on the outside, there are a whole bunch of things that get bound and recruited to the inside as well, right? And that allows the outside information, the fact that the cells are receptor is bound on the outside, to communicate inside the cell, and it talks to different things in the cells, and all those functions that we talked about earlier, right, that the matrix regulates, is controlled by everything that is happening inside the cell, right? And this is how the integrin molecule looks like, right? And this is just to kind of give you a sense of the fact that, okay, these are real proteins. They have a lot of information is known about the architecture of these proteins. And just one point I want you to look at and see how, the fact that this is a receptor that has, a region that's on the outside, which is called the extracellular domain, and a region on the inside. Can you also see that region that's in the membrane, right? The kind of spiral thing that is sitting in the membrane. That's an interesting part of that protein too, because that's what is keeping it in the membrane, right? And so, like all other proteins, they have very complex structures, and they do very intricate things. And a lot of this is studied, right? We know how these proteins work as well. The same kind of thing happens in cell-cell junctions. Okay, and now there is another protein that kind of comes this way to hold things together, right? And this is a class of protein that are called cadherins, right? And they actually, like a zipper, attach to each other and work with each other, and allowing cells to be brought together. So just like as I told you about endothelial cells that line blood vessels, even the cells on your skin that are kind of attached to each other, these kind of integrin molecules, the fact that they are able to come and bind to each other is what's allowing for that to take place. Okay, and that's what these cadherin molecules look like, right? They are the zippers, the equivalent of zippers in cells per se, right? The other interesting thought that I wanted to introduce here is that, I've told you how these architectures are created. I've shown you how tissues like the brain and the heart look like. One of the things that you should think about, right, and this is important because also of the extracellular matrix, is that these organs, right, also have a very distinctly unique property that is captured in this image, right? Now, when you walk on sand and when you walk on a hard surface like the brick here, what you immediately notice is how one is softer and one is stiffer, right? And your body kind of adapts to that, right? So running on a hard surface, running on sand is actually harder. A lot of athletes, for example, train in sand simply because that requires a lot more energy, and it kind of builds muscle strength in ways that otherwise is not possible. Similar to that, this matrix that we are looking at in cells, in tissues, also varies in stiffness. So along with creating shape architecture, it also creates an environment that has a very distinct stiffness, right? So the brain, for example, has a certain stiffness, the bone has a certain stiffness, and this stiffness varies very dramatically among our organs. And the reason this stiffness is able to vary is because of the extracellular matrix, right? Because of this mesh that now is made up of all these little proteins that come together. The way they are brought together and put together determines not only the composition and the behavior of cells, but it also contributes to how soft or stiff that matrix is going to be, right? So that's another important thing to consider when you think about organs and tissues, right? Not only do they have characteristic shape, not only are they made up of very unique cells, but they also have very unique stiffnesses. Okay, and in disease conditions, for example, that stiffness actually changes. Okay, which is another interesting thought. So as I said, the behavior of cells will change depending upon whether they are on a soft or stiff matrix. So when I told you about taking kidney cells and putting them in the matrix of the brain, at that point we were thinking about the composition, but along with the composition, the matrix could also have a very unique stiffness. So one of the reasons these cells don't do well there, or the cells don't do well here, is because of the composition and because of the stiffness that composition creates as well, right? So this then brings us to the all important questions of question of what happens if these cells don't stick, right? And there are many disease conditions that are created, where the cells actually don't stick, if the cells don't stick, right? And this include diseases like autoimmune diseases, diseases like cancer. When I told you about cells from the liver not being able to survive in another organ, now you think about diseases like cancer, right? If there is a cancer of the liver, what happens that also contributes to the deadly nature of many cancers is their ability to do something which is called metastasis, right? And some of you may have heard of this stuff. If you haven't, go look this up. It's the ability of these cells to go to new places and make tumors in new places, right? And that ability is mediated by the fact that these cells have undergone change to become cancerous, and when they undergo change to become cancerous, they don't seem to care what their microenvironment is. They are not regulated by the matrix as well as they were before, which is why they can go to new places, which is why they can grow and make tumors, and they can make tumors in new places, very different from their original tissue, right? The other example of how cellular behavior can regulate a disease condition is something called atherosclerosis, which is what I have highlighted here, right? Now, you all know what atherosclerosis is. It's just that you haven't heard of this particular term, right? The way you've heard about atherosclerosis is what's called a heart attack, right? And almost everybody knows somebody who at some point of time may have had a heart attack, right? And the question, many people can die as a result of the heart attack, but many also survive a heart attack and recover, right? Atherosclerosis is what happens that leads to a heart attack, right? And the video I'm going to show you next, and there is an audio that goes with it, and I hope you will be able to hear the audio as well, describes what atherosclerosis is. It will show you blood vessels and how in the blood vessels, these cells that I told you that line the blood vessels and the cells that are around the lining, how all of them contribute to actually come and create a block in the blood vessel, Okay, so a good part of what happens in a heart attack is one or more blood vessels in your body, which normally should be open and there should be nice blood flow through it, actually slowly get blocked, right? And to get to a point where it's completely blocked, okay, because of which blood flow is affected, right? And it happens around the heart more often than other places. And that ensures that the tissue around that blood vessel starts dying, which is why there is an effect where the heart loses its regularity, and in some cases can even stop, right? So the video we'll watch first, and that will make things a lot more clearer. Okay. Now, as I told you, that a lot of this has to do with this blood vessel, and as you can see, the blood vessel is actually made up of many different layers. The endothelium is the innermost layer, and there are all kinds of cells that make up that blood vessel. Remember, the blood vessel is a very elastic structure. Every time blood flows, it does this. Okay. Now, when you put your finger on the pulse and you can feel the pulse, okay, you're actually feeling the blood vessel, and you're also feeling the ability of the blood vessel to do this, right? Expand, contract, expand, contract. Every time there is a gush of blood that's going through that, right? And that's possible because this blood vessel is elastic. And that elastic nature of the blood vessel, ability to expand and contract, is driven by the matrix that's surrounding the cells. Okay. And this can get blocked, right? And this is like a tunnel that gets blocked. And when that gets blocked, it leads to something called an atherosclerotic plaque. Okay, you don't have to worry about the names. I've just added it there so that you know what it is. And it's actually a group of cells that are actually coming and blocking this blood vessel. And that's what leads to a blockage of blood flow and eventually the heart attack. So, if you have to, restore blood flow, you have to find some way to kind of clear that blockage. And I'll show you later how that clearing of blockage happens. Okay, so we're going to watch the movie, and let me know if you can hear it.
Atherosclerosis is a life-threatening disease that may have begun to develop during childhood. This condition is a process in which deposits of fatty material, called plaque, build up inside the walls of arteries, reducing or completely blocking blood flow. Although the exact causes of atherosclerosis are not clear, many scientists think it begins with damage to the inner wall of an artery, called the endothelium. Substances traveling in the blood, such as cholesterol, fats, and cellular waste products, accumulate inside the damaged area of the arterial wall. Chemical reactions occurring within the buildup of material cause cholesterol molecules to oxidize. This initiates an inflammatory response in which the endothelial cells at the damaged site release chemicals that signal a call for help. In response, monocytes from the bloodstream travel to the damaged site. Stimulation from oxidized cholesterol converts the monocytes into macrophages. The macrophages eat and digest the cholesterol molecules. As a result of this process, the macrophages change into foam cells, which accumulate to form plaque. As the plaque increases in size, the arterial wall thickens and hardens. At the same time, smooth muscle cells within the arterial wall begin to multiply. Most of the smooth muscle cells move to the surface of the plaque. These cells contribute to the formation of a firm, fibrous cap covering the plaque. Eventually, the passageway through the artery narrows enough to reduce blood flow and the amount of oxygen received by the organs it supplies. Over time, the cap may erode and break open, releasing plaque into the bloodstream. The plaque can flow downstream and contribute to the formation of a blood clot, which can stop blood flow. As a result, limited blood supply is available to the area surrounding the partially blocked artery. degrading and potentially killing the neighboring tissue.
And that's what eventually leads to a heart attack, right? So this is everything that we kind of talked about. I'm thinking there are a lot of small aspects that you would have seen in the movie, right? You can see the endothelial cells, the coverage around it, how the vessel looks, how you can see the matrix mesh that I talked about, right? This Maggie of life that's right next door, and how all of this comes together to block an artery and eventually lead to a heart attack, right? And now if you have to fix this thing, the only way to do this is to go in and kind of open the blocked artery, right? And they do this with something called a stent, which is actually very interesting because it's a grill. It's like a mesh, right? That is made up of titanium generally, which you carry in a small tube, which contains a balloon, and you move this tube through a catheter into the place where exactly there is a block. So you bring it there, and then what you do is you inflate the balloon, right? So you can see the balloon inflating, and what the balloon does when it inflates is that it opens this grill. Okay, the grill is designed in such a way that once it opens, it doesn't close. Okay, which is such a smart thing to do, right? Because once it opens and it takes its full shape, it ensures that it stays open, and that ensures that the blood vessel is actually now kept open. And now what they are doing is that these stents that are put in, right, these grills that we put in place are coated with extracellular matrix proteins, the same matrix proteins, that surround the cell. You take matrix proteins that are actually beneficial for the cells, proteins that actually will support the staying there of good healthy cells, and not allow for cells that actually can create a new plaque to come and accumulate there. So they coat the stent, and then take it in and then open it, right? And this is what generally happens in this procedure, which is which is called angiography and angioplasty, where they actually open blood vessels. Now, in some cases, the blood vessels cannot be opened, and what they do is they take that blood vessel out and they kind of connect the other two sides of the blood vessel, removing this portion, and that's what's called a bypass surgery, right? And that essentially allows the blood vessel to now work, because the portion that is damaged has actually been removed, right? So all of this is governed by this extracellular matrix, right? And I've shared this this poster, and we will make it available to all of you as well. This is a poster that we put together for an article on the matrix in a very interesting magazine called I Wonder. Okay, and I pitch the I Wonder magazine every time I speak at Inspire Camps, because this is a magazine that is produced by Azim Premji University. It's something that a lot of scientists from across the country contribute to. It's available online for free. And if you write to them and subscribe, they also send you a hard copy, right, to your library, to your school library, and you can request for one of those as well, right? And it's a very interesting magazine. It's published every 3 months, and it's now available in multiple languages. Okay, it used to be only in English. It's now available in Kannada. It's also available in Hindi. We wrote an article about the extracellular matrix, about everything that I just told you about in this, and I'll share the article as a PDF file along with the link to the poster as well, and you can look this up. Okay, I'm going to take the last about 5 minutes. Give me 5 more minutes to show you something that we do in the lab. Right. And now that you know the matrix, I'm going to introduce you to this idea of 2D versus 3D. I think this is an idea that we are all familiar with in some form or the other. A lot of cells that we grow in the lab, and I showed you an image or movie before, is cells that we grow on plastic dishes, right? So these are very hard surfaces. They are flat surfaces, and the cells stick and attach and spread and do everything that they are supposed to do. But in physiology, your brain is not a flat, hard surface, is it? Right? It's a three-dimensional structure, right? And so, the question is, you take cells and you put them in plastic dishes like this and you study them. But how do you know what the cell is doing here and what the cell is actually doing in the brain, which is a three-dimensional structure, whether that is similar? It's very unlikely it is similar, right? Because this is a very different environment. It has a very different stiffness, and it's flat, right? Which is not the case here. And so scientists over many years have understood this and have been trying to determine how cellular behavior is similar or different in 2D versus 3D, right? So you put cells on flat surfaces, this is what they look. In 3D, the cells begin to begin with itself, look very different, right? And they function also very differently. So one of the things my lab does, because we study cell adhesion, a lot of our experiments are done on flat dishes, right? And we put cells on top and we watch what they do. But we also look at cells in three-dimensional environments, asking, how the three-dimensional microenvironment works, right? So the two-dimensional, cells are looked at on slides like you probably handled, and for 3D we use something called chambers, right? And these chambers are interesting because they have a bottom which is made up of glass, and then they have chambers on on top in which we can grow cells. And the way we do this is we put a gel of collagen. So the pink thing that you see there is actually a gel of collagen. On top of that, there is medium to keep the cells happy and healthy, and the cells are actually inside the gel, right? So unlike cells which are nicely stretched out, these cells are actually big balls which are making kind of protrusions. These are actual images that are taken of a cell in a three-dimensional collagen gel, right? And I'm going to show you a movie next. And a lot of this imaging we do with something called a laser confocal microscope. Now, the laser confocal microscope is slightly, is like all other microscopes. It allows you to magnify and see things, but it also does something even more interesting, which is, it gives us images, with a certain clarity or quality that a regular microscope is not able to do. And the reason it is able to do this, so these are two images that are taken actually in a regular microscope and a confocal microscope, and you can see what kind of clarity it is able to generate. And along with clarity, it is able to generate three-dimensionality in an image. Okay, so the way it images is, and my best way to explain this is, looking at a bread loaf, right? And we've all handled a bread loaf, and you can, you have slices of bread as well. Now, imagine if you can take a cell and make slices of it, right? With the images that you take, right? And then you take all these slices, the order in which the slices are taken, and you put all of them together to see the full structure of the cell. Right? That's what we are trying to do with the confocal microscope. The above image that you see is actually of pollen, right? Which plants make. And, in the confocal, we take optical sections, right? So this pollen is actually not being cut physically. It's being cut optically, and every slice you take an image, and then you put all the images together to create this image of a three-dimensional pollen, right? And we do this for cells using a confocal microscope. This is an actual image of a cell expressing a protein which is in a three-dimensional gel, and we've done exactly what I just showed you, right? Which is we took sections of the cell and then we put all the sections together, and now we can rotate the cell in all directions and look at it like a three-dimensional structure. You can see the kind of protrusions it is making. You can see how hollow it is. You can see, what thickness these protrusions have, all of this, right? We can also do imaging where we look at the movement of live things inside the cell, right? So we take a protein and we put a fluorescent tag to it, something that will kind of shine light, right? The moment we shine laser on this, it makes fluorescence of a particular color. In this case, it is green, right? And so we can now watch where these molecules are moving in the cell in a three-dimensional space, right? So not only are we looking at cells, we are looking at things happening inside the cell as well, right? Which is quite remarkable for us to be able to see. And then we are able to do all of this in 3D. right? Which means we are able to take sections and we are able to shoot movies of things that are happening inside the cell. So what you see there is a cell that is sitting inside a three-dimensional gel, and you can see how it is wriggling. Okay, this is a movie that is taken over maybe a couple of minutes, about 10 minutes or so. But you can see how the cell is wriggling, constantly changing its shape. You can also see, we are able to see things that are happening inside the cells as they are kept in a three-dimensional space, right? And this allows us to study the behavior of the cells with this environment that it is in, right? Remember, the outside of the cell is actually empty right now, right? But in this image, it is empty, but it actually has collagen. It has this mesh, and it allows us to see now the behavior of the cell in context of that matrix. We can change the composition of the matrix and look at the behavior of the cell. We can change the stiffness of the matrix and look at the behavior of the cell. All this is possible because of the kind of imaging that we are able to do, right? The images that I showed you are taken by Trupti and Vishakha. Trupti, who was a graduate student in our lab, and Vishakha, who was an undergraduate student. And the images that I've shown you were all taken by them. Once again, this is where I'm speaking to you from today, and a lot of the work that that my lab does is funded by many different funding agencies, which allows us to do all these cool experiments that we do. We have a website, and a Twitter handle that you can go, look us up on, right? The Twitter handle is at the rate Adhesion Lab, and the website has a lot of information about what we do, the things we do, right? And this is some of the people who do the work, right? And we have students from across the country, and all the way from Darjeeling, to the southern tip of India, right? And so it's a great environment. It's a great place, and to come and study and work. And I'm hopeful that some of you will aspire to be in a place like this, right? IISER is a very beautiful campus, a terrific infrastructure, and as somebody who's lived and worked abroad for many years, I can vouch for the fact that this is as good as anywhere else in the world, right? So I always am envious of my students, because I never got the opportunity to do this kind of science or do it in a place that has this kind of infrastructure in India. I had to go abroad to kind of experience something like this, but they have the opportunity to do this here in the country, as do you, right? So look us up. If you have questions, do come back. And there are links to some of the movies that I showed you today. I'm going to leave you with that last slide here of the lab, and I will be happy to take any questions that you may have. Right? I'm going to go to the chat box as well, see if there are any questions there, and try and see if I can answer them. Okay, so I'm going to go up to the first set of questions and see if we can go one at a time, right? I think you can see the questions as well. Somebody wanted me to speak in Hindi. Oh, I'm so terribly sorry. Okay. Yeah, you can ask me a question in Hindi and I will be happy to answer it, right? But I think some of this is difficult to communicate for me also in Hindi or in Marathi. I do speak a bit of Marathi. So if you have a question later on that you want to ask after we take these questions, you can put your hand up and I'll try and answer it as well, right? So I'll take some of these questions. Is the matrix there in every organ? Yes, it is. Okay, that's a good question. It's there. Yes, when I was talking about the membrane, I meant the phospholipid membrane. You're right. Yeah, so somebody's mentioned that they looked at Velcro under the microscope. So then you know what I'm talking about, right? And if you haven't, even if you take a magnifying glass, it doesn't need a microscope. Look it up with a magnifying glass, and you will be able to see how Velcro works. And it's really remarkable as an idea, and it's also very interesting how many of these ideas have their origins in biological systems, right? Like we seem to be trying to do what our bodies and cells and tissues do really well, right? Can integrins be pulled apart? Yes, they can be pulled apart. So every time a cell moves forward and detaches, there is actually physical force that is applied to kind of detach the cell. Okay, so it's actually pulled to to detach the integrin from the matrix, right? Can atherosclerosis be caused by obesity? Obesity is a contributing factor. It's not the only thing that can cause this, but it is indeed one of the contributing factors that can lead to atherosclerosis, right? One of the questions is that is it possible that a stent is inflated too much and it bursts the blood vessel? See, the stent is designed in such a way that, as I said, it opens to a defined size and then doesn't close. Okay, so it's not a stent that will keep on opening as much as you push it, right? So it opens to a specific size and then it will not close back, and that ensures that you don't do what you are thinking, right? The cells, I think you're asking me whether the things that are fluorescent inside the cell can be red in color. Yes. There are many different colors that you can make now, and it's very interesting. The protein that is used to actually label proteins inside the cell is this fluorescent protein called green fluorescent protein, GFP, right? And it's actually coming from a sea animal, right? Which naturally makes this protein. It was discovered there, and they said, oh, these sea creatures make this bright green fluorescent color, and they found what is it that they are using to make that color. And it was a very smart thing to be able to take just that and put it in another protein. And what do you know? You now have a protein that is fluorescent, like this, like these proteins in these sea creatures were. And now you can watch where the protein goes inside the cell, right? Which is a very remarkable thing, right? Okay. Yes, they can grow in red. Also, there are now people have done very smart things. They've looked at this GFP protein and they have figured out what exactly gives rise to the color, and they have made small changes in this protein to now create versions that make light of different colors. Right. And so this now allows you to look at 2 proteins or 3 proteins, sometimes in the same cell as well. Right. Are there measures to prevent damage to a matrix? Very good question, right? And this is something to think about. So a lot of, like all other organs in our body, I think what we put into our body obviously influences everything. Right. So the kind of food we eat, the kind of exercise that we have influences the tissues, the cells, and also determines the overall health of your body. Right. So clearly, the health of the organs is affected by the matrix. And every time you exercise, for example, I think the fact that the muscles in your body or the heart is exercised the way it is, among the things that it is doing is also affecting the extracellular matrix, right? So it's hugely important that you maintain as much as possible a healthy lifestyle, because that does contribute, among other things, to the health of the matrix, and the health of the matrix will then eventually determine the health of your cells, which together will determine the health of your tissue and organ and your body per se, right? So yes, it's important to take care of your matrix as well, right? Can stress cause a heart attack? So stress is a contributing factor. Yes, it's known, and some of that has to do with what kind of impact stress has on your body, right? And this is again a very diverse and complex area, and I don't want to get into everything about how stress affects your body, but it's documented very clearly that it does. And among the things it does is affect the behavior of different tissues and organs, right? And that includes the heart as well, right? So just as cells can capture bacteria, they can also capture viruses. The viruses are more challenging simply because of the numbers, right? And viruses are more challenging also because they can undergo mutations, and now we know what mutations mean, right? We know how there are all these variants of this particular COVID virus that we are all now familiar with, right? There is this Omicron variant now, right? And so viruses keep changing, and which is why it is very difficult. But macrophages, are able to eat up viruses like they are able to eat up, bacteria as well, right? Somebody's asked me where ICER is. ICER is in Pashan. Right, we are very close to, we are on the Pashan Sus road. So if you know where the DRDO ARDE is, or where National Chemical Laboratory is, we are right next door to it, right? And right now, you can come and visit. I think it's not difficult. I think the campus is open, and so you can come one day and just say that you wanted to go see the campus, and chances are they will allow you. If they don't, schedule an appointment with somebody that you want to meet, and you can come and see the campus that way as well, right? Can the blood vessels dilate or contract if this is helped by a stent? Yes. So it will be compromised, right? But that's why the stent is a very small part of that blood vessel. So, so you're right. If the stent is there, the blood vessel is not contracting, right? And so that will affect the behavior of the blood vessel around the stent. But you're kind of thinking it's okay for that to happen as long as the blood vessel stays open, because the objective here is to kind of clear that blood vessel and open it per se, right? So you're right that region will not do this like the rest of the blood vessel, but you're taking that chance. You're thinking that's okay, considering what it is allowing you to do, right? Somebody has asked me a question saying they're interested in research, and she stays in Pune and don't know much about ICER. Look up the ICER website. There is a lot of information about ICERs. Look up the labs that are present here. Every lab has a website, including mine, right? And we tell you about the work that we do. Many labs are also actively engaged in communicating the science that they do. Like I gave this talk, there are other people who will be speaking at different times. The ICER also has a science activity center, okay? And this is something that you guys should all check out. They every week they conduct live tutorials. They have a YouTube channel in which you can go watch this, and there are videos from past tutorials. This is also a nice way to stay connected to ICER, because every week you can subscribe for this, and they will tell you what the next tutorial is going to be on. There are scientists who come and speak there, and this happens every week, and or every 2 weeks, I think. And this is something that is open to all. And now, because of Zoom and other things, a lot of these talks are broadcast live on YouTube, right? So you should go check that out as well, right? And if you're interested, look up the website, write to labs. There are students who come and spend small amount of time in the lab, even 10th and 12th standard students who have come to my lab and spent like 3 weeks, watching the kind of research work that happens, right? And so we are open to this possibility, not just my lab, but other labs. And so you can, write to labs, right? Soham has a question. So you said that matrix is different in different cells. So the composition of the 4 proteins changes in each cells. The way the cells are able to secrete these proteins changes, which means that in the matrix, the relative amount of these 4 proteins could be different. Remember, it's not 4, okay? This is just the tip of the iceberg. There are about a few hundred proteins that make up the matrix, okay? And the composition of each of these changes depending upon the tissue. So it's a really complex structure in that sense. And I kind of simplified it to allow you to understand it, but it's much more complex than that, right? Atherosclerosis leads to myocardial infarction. That's correct, right? So you're right about that, Soham. Do you have to give a special exam to get into ICER after a 12th, right? So there is an examination, like the IITs, there is a national entrance, and you can go look up the ICER website. ICER Pune website will also tell you, have a link to the ICER entrance website, and you can find out when the entrances are held. And as I said, students come to us from across the country. Students take this examination. They take sometimes the KVPY examination. There are multiple ways by which you can gain entry into ICERs. ICER is also has an integrated PhD program along with the PhD program. The integrated PhD program allows you to come and join after a BSc, right, to a place like ICER, right? So look us up, and there is a lot of information online on the ICER website. If you haven't been to the ICER Pune website, go check it out, right? It's icerpune.ac.in, right? A lot of debate about whether the viruses are alive or dead. If they can divide and replicate, I'm tempted to say that they're alive, right? And the fact that, but you're right, there is a lot of debate about it. I think what probably matters is what they do. You can consider them dead, but if they are able to divide, replicate, spread, you still have to be worried about them, right? So I think, again, it depends on whom you're asking here. But for me, the fact that they're able to replicate and they're able to make copies of themselves, can let you classify them as being living things. As I said, that's a loaded statement, so don't hold me to it. It depends on whom you're asking as well, right? Which molecules stabilize the extracellular matrix and give it the ability to resist compressive forces, right? So the matrix in itself, okay, all these 4 molecules and all the other molecules that are part of the matrix contribute to regulating the architecture of the matrix, right? So along with the big fibers, they also regulate kind of cross-linking of these fibers, and that in turn determines, how soft or stiff this matrix is. It also determines the shape of the matrix. So all these proteins contribute to the architecture of the matrix, and that in turn regulates the ability of cells to resist the kind of forces that it experiences, right? All about ICERs can be discussed later, that's fine. Okay, so I think, the question about ICERs, I'll be happy to take at the end. If you have questions about the matrix and what we talked about today, maybe we'll take that first, right? And whoever is coordinating it, I'm sorry, I didn't catch your name. Let us know how we should proceed forward, and I'll be happy to take questions about ICER as well. Whenever we are ready to do so, right? Any more questions? Anybody wanted to kind of put their hand up and ask a question, or yes, somebody has their hand up. Okay, go ahead. You can put your hand up and then ask the question. I think that'll make it easier for others.
Yeah, we would request all the students to ask sir questions related to the topic of his speech, because general questions about how to get into ICER and other things can be, or a separate session is arranged on the 26th of December for the same. Okay.
Perfect. Yeah. So you will get to know, but I think it's important for you guys to know. I think that's the first step to kind of considering a place like this as well, right? Yeah. So any questions on what I spoke about, please go ahead. I'll be happy to take them.
Yeah, because about ICER and the different exams and all the session will be taken by Professor Rajan Waldve sir and also by Dr. Arvind Nathu sir on the 2nd of January. So we'll be having a very clear picture. Yes, students, any other doubts regarding today's topic? Yes, Samruddhi has raised her hand. Yes, Samruddhi.
Please go ahead. Yeah, go ahead.
You can also unmute yourself and speak. Yes, Samruddhi.
Yes, sir, in the PPT you mentioned white ghost heart. So can you tell more about that?
So it is essentially, there are methods by which you can take a tissue or an organ and you can completely remove all the cells from it, right? So it is processed in a very specific way such that only the cellular material is taken out, right? And so what is left back is essentially the extracellular matrix. And if you do that to the entire heart, right? You treat it and process it in such a way to get rid of cells. What you're left with is this ghost of a heart, right? And that ghost of a heart is actually nothing but the extracellular matrix that the cells have made, during the lifetime of the cells, during development, that has created the architecture and the shape of the heart, right? And what that is telling you is that the shape is not lost when you remove cells, okay? Because the shape is actually coming from the matrix, which the cells make, yes, but the cells are not responsible for the shape. The matrix is, right? And that's probably true for other organs as well, like liver and kidney. And this is another important thing to remember, that right, different organs have very distinct shape, right? And they have very distinct stiffness. I don't know whether, not many of us get an opportunity to kind of hold an organ in our hand, but if you hold a liver and you hold a brain, you can feel the softness or stiffness of these tissues as being different, right? And so they have very distinct shape, they have very distinct stiffness, and both of these are contributed by the extracellular matrix. And that's what removing of the cells is showing us when you look at that heart, right?
So, sir, this white ghost heart is used for experimental purpose.
So it is, you see, what they do is that, you can take now, essentially what is left back is the matrix, right? So they do use the matrix that is coming from that tissue. So if you want to make matrix from the heart, right, and not the individual matrix components, but just the complete matrix, you can do this. You can solubilize the cells and then take the heart and essentially break up the matrix now to create a powder of the matrix that's actually matrix coming from the heart, right? And so this now is many different matrix proteins, but you know that they're all associated with the heart. And this is done for different organs, which is why that process is actually is created, to allow you to make matrix that is coming from that particular tissue, right?
Yeah.
Okay.
Yes, sir. Thank you.
Okay. Somebody had a question in Marathi. मराठीमध्ये विचारलं तर चालेल. माझी मराठी एवढी चांगली नाहीये, पण मी प्रयत्न करणार. Okay. Okay, the next speaker is scheduled at 12. So I think we're almost out of time, right? We have a few more minutes, and I can take a few more questions. Is that okay?
Yeah, definitely, sir.
Okay. How exactly is the integrin reacting with the collagen matrix? It's a good question, Vaishnavi, right? So these are receptors, right? So they actually go and bind, right? So there are things in the extracellular matrix that this receptor, the integrin receptor, actually has affinity for. And that affinity is coming from, the interaction. See, this is also protein. So it's made up of amino acids. This is also protein that's made up of amino acids. And this has a particular architecture. This has a particular architecture. It goes, sits, and when it sits, it can bind, with a high amount of affinity. And that is what ensures that, this is able to kind of hold on to that. Now, when that happens, what is interesting is that the inside of the receptor actually undergoes change. And that tells the receptor, the cell, that on the inside, that there is binding on the outside. Okay. So a lot of that happens by covalent interactions between amino acids. Okay. Okay. Any other queries? Okay. How does the microscope know where the bacteria is? The microscope doesn't know where the bacteria is. The person looking through the microscope knows where the bacteria is. That is why the microscope is moved. The slide is moved in such a way that you can follow the bacteria. Okay.
I think he has written macrophage. I think he means.
Oh, macrophage. Sorry. Okay. The macrophage, that's a very, very interesting question. Okay. So how does the macrophage know the bacteria, where the bacteria is? So the macrophage has receptors on the surface. Okay. And then just imagine this, right? Just as it has integrins, it has other receptors on the surface. And these receptors actually bind to molecules that the bacteria releases. So when the bacteria moves from this position to this position, the release or the of these molecules also moves the bacteria. Right? So the bacteria now is releasing, these chemicals, which are called chemoattractants, from this particular site. So from this site, it moved here. And what happens is when the cell is sensing these chemoattractants, initially it was looking at a lot of chemoattractants in this position, right? Because all the receptors that are in this area are actually binding to the chemoattractant. But now, when the cell has, the bacteria has moved, the chemoattractant signal has moved, right? So the receptors are now bound more in this particular site, and this the cell is able to sense. So it's able to say, okay, a lot of receptors are bound here. I need to go in this direction. When the bacteria moves, now the receptor binding has changed. So it knows that the chemoattractant is not coming from this place, but from this place. And it changes its direction on the basis of this, which is a really remarkable and smart thing to do, right? And it's a complex thing to do, but the cells are able to do this, right? So what happens to the unfortunate bacteria? The bacteria generally, once it's engulfed, there are enzymes that are inside the cell, things that are carried around in bags, which are called lysosomes, for example. They generally come and fuse with this vesicle in which the bacteria is, and they actually digest the bacteria, right? So the bacteria gets killed inside the cell, right? So when a disease is not cured, Okay, disease is not cured. Can we say that the macrophages are weak? So it depends on the disease, right? So something like, for example, the cold, if it is taking longer to cure, okay, it's probably because your immunity is low, and because the number of bacteria that are, for example, could be higher, and so the macrophages are trying to fight it, but they are not able to kill it very well. Remember, the macrophages actually end up dying as they eat bacteria and kill bacteria, right? So for example, when you have a cold, and the phlegm that you make is actually dead macrophages, okay, which are trying to fight the infection, right? And that's an indication that your body is actually trying to resist, and that's why the general immunity that you have matters, right? But sometimes, if you're unwell with a very severe infection, you need drugs to kind of recover, and that's probably because the macrophages are not able to deal with the amount of the infection that you have, right? Okay. The macrophage can be overwhelmed. Absolutely, that's possible, right? So that can happen at times. Samruddhi, you raised your hand again.
Sir, I have one question.
Sure, go for it.
I have one question, that is, there is any character of cell organism in matrix or not?
So when you say character of the cell or cell in the matrix, what do you mean character? See, the matrix is made by the cells. Could you repeat that again?
That there is any character of cell organisms in, means organ of that cells.
So organ, I'm not sure I understand the question completely, but if you're asking if the matrix is in a particular organ, okay, is actually unique to that particular organ, the answer is yes. Right? So if you take, like, if you had a way to kind of look at the composition of the matrix, I did not tell you where this matrix is coming from. I just gave you a chunk of matrix and said, this is matrix from some organ. Now tell me which organ, right? If you can analyze the matrix from different organs, what you will know is that each organ has a very distinct composition. So if you look at the composition, if you look at a piece of matrix, you will be able to tell which organ it came from. And that's happening because the cells make this very specifically in that particular organ.
Okay, sir.
Thank you. At what temperature do extracellular matrix proteins denature? So see, a lot of what these matrix do is at the temperature that our body is at, right? So 37 is optimal for them, right? And it takes much longer for these proteins to denature, you have to go up to 90 degrees, for example, to be able to do this. Right, 90, 100 degrees, they will start breaking down. Some are fairly stable, okay, and will last even if you go to those temperatures. Their architecture may change, but they will not completely break down. So cells can be frozen, yes, you put them at 4 degrees, they will get cold. You take them to 0 degrees, they will freeze, right? The cell is made up of has a lot of water in it, so how you freeze them will matter, right? And if you freeze them very quickly, this water does not crystallize very much, and so the cells kind of are frozen without actually too much damage, right? But if the cells are frozen slowly, the water kind of crystallizes and makes ice structures, ice-like structures, and which can poke and damage the cell and actually destroy the cell. Can macrophage call for reinforcement? Yes, it actually does. Macrophages will call other cells. When a macrophage dies, for example, because it has eaten too much bacteria, the things that it releases now calls more macrophages there, right? Which is why, for example, if you have a cut in your hand and you see, the cut healing, watch it carefully next time, right? You can see, for example, some, oozing that happens there, which is actually coming because these cells have all gotten there and they are trying to fight whatever bacteria have gotten in through your skin, right? So the macrophages do call for reinforcement, right? So cells, can they be revived, right? And that is an interesting question, because depending upon how you freeze them, there are some cells you want to revive later, right? And if you freeze them carefully, if you freeze them in a medium in which the cells are kept healthy, you can actually revive cells after freezing, right? And they will recover and grow normally. We do this all the time in the lab, right? And so it's possible, right? Birthmarks, yeah, probably. Sure, I think any mark that appears on your skin, if the birthmark is on your skin, I have a birthmark here on my skin, this one, that's cells, which have a certain pigmentation, and that's been there, and it stays. So yeah, so certain discoloration of cells can cause these marks, absolutely, right? Yeah, so how do you extract matrix from tissues? So there are methods to do this, right? And there are ways to solubilize the cells, get rid of all the cells, and then kind of only keep the matrix. And there are labs that do this, there are companies that do this, right? And it is something that is considered a very useful thing to do. I mean, you can't do this from human tissue yet, right? Because there are challenges to that. So they do this from other tissues, like from pigs. A lot of the matrix is generated, right, which are grown, just for this particular purpose.
Yes.
Okay. We're almost 12 o'clock. Maybe we should let the next speaker come in and talk as well. I'm sure they have interesting things to say as well. Yeah. So back to you.
Yes.
Yes.
Yes, Soham.
You told that some bacteria can be revived if they are freeze. So,
No,
sir,
cells can be revived. Cells can be revived.
oh yeah.
Bacteria also revive, by the way. Okay, that's also fair.
Is it possible that there was an anthrax outbreak in a certain area, and then it got buried by snow, and then the snow melted, and the anthrax made a resurgence?
Possible, right? The bacteria can go and survive in many different places as well. It doesn't have to be only under the snow, right? So they could be stored, or they could be trapped in certain places, which allows them to come back later when the conditions are better. Yeah, they do this all the time.
Thank you, sir.
Sure.
Yes, that was such a patient session by you, sir. And it almost left me with the feeling that you personified every aspect of the cell as if we are talking to them and understanding them. Thank you so much for your time. Thank you so much for all the, answering all the questions on behalf of the trust. And we look forward for such camps in the consecutive years, and hope that you will always have your blessings for the same, sir.
Yes. It was wonderful to meet everybody. Right. I know I have not seen any of you. Right. And this is one of the challenges that of giving a virtual talk is that you don't get to meet people in person. Right. But it was wonderful to be here. And thank you again for organizing the camp. And good luck to all of you. Good luck to all of you.
Thank you so much, sir. And as per your input, if we plan a trip in ISA and if you are not allowed, we'll definitely connect with you.
Anytime.
And we'll love to see your lab. Students,
Sure,
students are also requested they can put their videos on so that the speaker knows whom the speaker is
talking
to.
If you can turn your video on, at least I'll get to see all of you once. Right?
Yes, and beautiful doubts by all the students. So I am sure the session was interesting. And also for sir, you to know after the session, like I also have been making some notes. There are asked some questions. There are some certificates based on the sessions and the questionnaire, which we'll be putting forth after 1:15.
Okay.
Right, so students are attentively, listen to the sessions. It's not just like you put on the Zoom and up se sun rahe ho. Right. So once again,
and ask
you,
questions.
Yeah.
I will urge all of you to ask questions. Don't be scared of asking questions. It's okay if you're wrong. It's okay if it is sometimes difficult to properly communicate what you want to ask, but ask. Okay. Don't ever stop short of asking questions. At your age, the more questions you ask, the better you are off. Okay. Good luck, guys.
Yeah, sure. And that is what, searching for questions, searching for answers again and again is researching, right? And so when the students ask how to become a researcher, just keep on searching, and that comes with questions.
Yeah. True.
Yeah, fine.
Okay.
Thank you once again, sir. Have a nice time, and we'll connect with you soon for such camps. Right?
Sure.
Yeah.
Thank you. See you. Bye, everybody.
For all the students, stay tuned here. You will have a break for 5 minutes. We were supposed to start with the next speaker at 12 o'clock, but you all had your brilliant questions and we could not stop them for sure. Professor Anisa Chorwadwala, who will be speaking on maths. She's also from ISA Pune, and her topic is why are soap bubbles almost spherical? Okay, so she will be explaining you some isoperimetric problems, which is really interesting. I have gone through the abstract and the research paper as well. So you can ask your friends if they want to join, they can definitely join. You are getting a break for 5 minutes and then we'll start with the next session. So stay tuned. I'll be back by 12:15 students. We'll take a break.